Welcome back. In the last lesson, we learned what messenger RNAs are and their general makeup. In this lesson, we'll learn how mRNAs function as the blueprints for proteins. Please take a moment to review the learning objectives for this lesson. Hopefully, you'll remember that the M in mRNA stands for messenger. These messages are in the form of the genetic code. What is a code? A code is a set of symbols that can be variously arranged in a linear sequence to convey information. One example is the binary code used by computers, which consists of only two symbols, 1 and 0. Other examples are written human languages like Greek and English. Whereas written Greek uses 24 different symbols or letters, English has 26. In both languages, these letters can be variously arranged to form words and sentences. We've already learned that messenger RNAs are long chains of 4 building blocks, A, C, G, and U. Which of these building blocks or nucleotides are present, and the order in which they are arranged along the chain forms a 4-letter language, the genetic code, or the language of life. Just like something written in binary computer code or Greek needs to be translated to be understood by someone who can only read English, the 4-part genetic code of mRNAs needs to be translated into the 20-part amino acid code of proteins. Thus, the biological process of making proteins using instructions encoded in mRNA is called translation. Now, let's learn about the cellular machinery that performs this translation process, the ribosome. The tiny molecular machine that reads the information stored in mRNA and translates it into a protein is called a ribosome. A ribosome moves along the mRNA and spits out the new or nascent protein. Thus, ribosomes are your internal protein factories. Here, we're watching one ribosome manufacturing one protein on one mRNA molecule. But really, this scene is more realistic of what is actually going on inside your cells, with many, many ribosomes working on lots of mRNAs simultaneously. Let's pause here and watch for a while. Remarkably, in order to make all the proteins your body requires, each one of your cells contains millions of ribosomes, hard at work manufacturing billions of protein molecules from hundreds of thousands of mRNA molecules. Remember that your body is composed of over 30 trillion cells. So let's do some math. 30 trillion cells times a million ribosomes per cell comes to 30 quintillion ribosomes. That's 30 followed by 18 zeros. Altogether, these 30 quintillion ribosomes are working on over a quintillion mRNA molecules. Wow, that is a lot going on. But how do ribosomes actually use the information in mRNA to create proteins? How do they know where to start, what amino acids to string together, and then when to stop? To understand this, let's go back and think about how information is encoded in written human languages. In English, the signal for the start of a sentence is a word starting with a capital letter. Individual words consist of varying groups of letters separated by spaces, and the end of a sentence is denoted by a period. However, in the language of mRNA, the genetic code, there are no capital and lowercase letters, there are no spaces, and there are no punctuation marks. Instead, the genetic code consists of the 64 3-letter words that can be made from all possible combinations of A, C, G, and U. These 3-letter words are called codons, and 61 of them are used to code for different amino acids. Looking at this table, let's see how this works. Let's suppose that a particular protein requires the amino acid tryptophan, or TRIP for short. The sole mRNA codon for tryptophan is UGG. But if a valine, or VAL for short, is required, there are 4 different codons that all encode valine: GUU, GUC, GUA, and GUG. So just like synonyms in English, some mRNA codons are synonymous and have the same meaning when translated into the language of amino acids. The existence of these synonymous codons enables multiple different mRNA sequences to encode the same protein. The codon AUG specifies the amino acid methionine, abbreviated as MET, and also serves as a signal for where along the mRNA chain to initiate protein synthesis. 3 other codons, UAA, UGA, and UAG, signify where the ribosome should stop. That is, they act like a period punctuation mark indicating where the protein should end. Thus, AUG serves as an initiation codon, and UAA, UGA, and UAG serve as termination codons. Now that we understand the structure of the genetic code, we can learn how the translation process works. First, with the help of some accessory machinery not shown here, half of the ribosome, called the small subunit, binds to the beginning of the mRNA and scans along the strand until it encounters the first instance of the initiation codon AUG. Once it's found an initiation codon, the small subunit pauses and recruits the other half of the ribosome, the large subunit. Once the large subunit is in place, protein synthesis commences. The ribosome ratchets along the mRNA, one codon at a time, snapping the correct amino acids in place to manufacture the encoded protein. In human cells, a ribosome can string together about 60 amino acids per minute. This means that the average human protein takes just a few minutes to manufacture. When it reaches a termination codon, the ribosome releases the full-length polypeptide, which completes its folding to become a functional protein. After its work is done, the 2 ribosomal subunits dissociate and are ready to start the process all over again. Finally, if you remember, I told you before that each cell in the human body contains millions of ribosomes manufacturing proteins from hundreds of thousands of mRNA molecules. That means that there are about 10 times as many ribosomes per cell as there are mRNA molecules. So even though the movie we just watched showed only one ribosome translating an mRNA, in actuality there are usually multiple ribosomes moving along each mRNA at any one time. This famous electron micrograph captured an mRNA with over 30 ribosomes in transit. If you look closely at this amazingly detailed image, you can see the mRNA as the thread that connects all the ribosomes. The ribosomes necessarily stretch out the mRNA as they move along. You can also distinguish the small and large subunits of individual ribosomes. Further, you can tell that the ribosomes are moving left to right because the protein strands emerging from the ribosomes on the right are longer than the protein strands on the left. Finally, on the far right, you can even see the proteins starting to fold into their final 3D shapes. It is estimated that each mRNA molecule is translated hundreds to thousands of times, meaning that each individual mRNA molecule is used as the template for hundreds to thousands of protein molecules. Exactly how many protein molecules are made per mRNA depends on how rapidly the ribosomes are loaded, how fast they move along the mRNA, and how long the mRNA sticks around before it gets degraded. In the next lesson, we'll learn where mRNAs come from.